Have you ever wondered why you or someone you love may have a genetic illness or disease? Or perhaps if your children may be plagued with a genetic illness or disease? These questions may already have an answer, and this is due partly because of the completion of the Human Genome Project. Hello, I am Jameson Pittman, and I'm going to give a brief speech on what the Human Genome Project is. The Human Genome Project was completed in 2003, 2 years ahead of the 15-year planned project. But this project could not have been performed without the discovery of the double helix DNA molecule by Dr. James Watson and Francis Crick. Here's the model of the double DNA helix molecule. It's composed of a nitrogenous base in the center, followed by a pentose sugar and a phosphate group. The Human Genome Project was coordinated by the U.S. Department of Energy and the National Institutes of Health. The Human Genome Project's goal was to sequence the entire human genome and a few other organisms as well, such as the Escherichia coli, better known as E. coli, and Drosophila melanogaster, which is better known as the fruit fly. Now when I say genome, I mean all of the genes that make up all of the chromosomes which are in all of the cells of an organism. And a gene can be composed of many bases, as many as 3,000 bases, or a lot more than that, such as 2.4 million bases. And you can see here that the gene has its individual nucleotides which make up the chromosome. 99.9% of all the bases found in the human genome are the same. It is the 0.1% difference that makes us unique. This uniqueness can be the difference in eye color or facial features, or it could be the difference in having a genetic disease or not. Even though the entire human genome has been sequenced, there's still about 50% of the genes that have been sequenced for which their function is not known. All of these sequences have been stored in databases so that scientists around the world will have access and benefits to the knowledge gained by the Human Genome Project. There have been many benefits that have resulted from the Human Genome Project. One such benefit has been in molecular medicine. This has improved diagnosis of diseases, and genetic predispositions to diseases can be detected for some diseases. For example, in this diagram here, person 1 has the exact protein in which they need for their metabolomics to occur. Person 2 has a single nucleotide change which does not result in a codon change for the amino acid, and so the alanine amino acid is the same for person 1 and 2. But one single nucleotide change in person 3's DNA has resulted in a different codon, resulting in the translation of the amino acid lysine instead of arginine. This results in a defective structure of the protein, which allows them not to have the same chemical reactions occur in their body as persons 1 and 2. Drugs have been created based on this molecular information. Gene therapy and control systems may be used as drugs, and custom drugs have also been designed based on individual genetic profiles. One area that most people are familiar with deals with DNA identification because of our exposure of it through the news. DNA identification, also known as forensics, has benefited greatly from the Human Genome Project. DNA can identify potential suspects whose DNA may match evidence left at a crime scene. DNA can exonerate persons who are wrongly accused of a crime. DNA can be used to identify crime and catastrophe victims. DNA can be used to establish paternity and other family relationships. DNA can be used to identify endangered and protected species to assist wildlife officials in identifying poachers. DNA can detect bacteria and other organisms that may pollute the air, soil, water, and food. And DNA has also been used to match organ donors with recipients in transplant programs. There's also been benefits to agriculture, livestock breeding, and other bioprocesses from the Human Genome Project. DNA information assists in growing disease, insect, and drought-resistant crops. It has helped in breeding healthier, more productive, disease-resistant farm animals. It has also helped in growing more nutritious produce, and has been used to develop biopesticides. However, the Human Genome Project has created some concerns in ethical, legal, and social issues. Privacy and confidentiality of genetic information is one issue. Fairness in the use of the genetic information by insurers, employers, courts, schools, adoption agencies, and the military must be assured. If an insurer knows that you are predisposed to dying at the age of 35 because of a genetic illness, then they will be less likely to give you life insurance. Reproductive issues, including adequate and informed consent and the use of genetic information in reproductive decision making, must also be considered. There must be fairness in the access to advanced genomic technologies. Conceptual and philosophical implications regarding human responsibility, free will versus genetic determinism, and concepts of health and disease must be given consideration. Health and environmental issues concerning genetically modified foods and microbes are another area of concern. And commercialization of products, including property rights and accessibility of data and materials, is another area of concern. In conclusion, I hope that you now have a better understanding of the Human Genome Project, with its benefits and what the ethical, legal, and social issues may be. The Human Genome Project was a 15-year major joint research project that involved numerous scientists around the world and was coordinated by the U.S. Department of Energy and the National Institutes of Health. However, it finished 2 years early, making it a 13-year project. The human genome and a few other organisms as well were identified, sequences determined, and stored in databases. Information for the project was stored publicly so that scientists around the world could use this information. And there are many ethical, legal, and social issues that have been identified. Thank you. The references for this are from Koshland, D.E., 1989, which is the journal called Science, page 246, retrieved in 1989. The U.S. Department of Energy, Genome Programs, and Watson, J., and Crick, F., from 1953, a structure of deoxyribose nucleic acid and nucleic acid, in the journal Nature, page 171, and that was the April issue. Thank you.
I was present for this speech.
And I likewise was present for this speech.